Okay, so we are very fortunate to have Peter Pachmann from Maintrack to explain to us what is the basics of circulating tumor cells.
Okay, thank you very much.
What is it actually all about? What is circulating tumor cells?
Okay.
What are you doing?
So circulating tumor cells are cells that disseminate from solid tumors. 85% of all tumors are carcinomas from epithelial origin, so they are solid tumors. And even after surgery or radiation, tumor cells can remain in the patient's body. And chemotherapy or a therapy with any substance that helps the patient is targeted to attack the cells remaining in the body. Okay. So what you see here is you see a primary tumor over here, and the cells flowing into cells from the tumor disseminate and get into the bloodstream and can build metastasis somewhere out of the body, somewhere out of the original tissue, not out of the body, and build up new metastasis in distant places. The reason why we don't call them circulating tumor cells, but circulating epithelial tumor cells, is that all of the cells out of a solid tumor are epithelial tumor cells, are epithelial. We have a public interest of circulating tumor cells.
But before that, if I may ask you a question, yeah. Could you tell which type of cancer it is, or all you know is cancer? Could you say breast, colon, prostate?
So we do not have the ability to tell you what kind of cancer it is, because if you look at this picture, you can see that even if it's a breast cancer that disseminates cells and it's a metastasis in some distant tissue, it may be cells that are from breast cancer doing the metastasis, let's say in the liver or in the lung. So with looking at the cells, you can't even say what kind of tumor it is, but you can say what the origin of all of this tumor cells is that are in the blood.
Okay.
So basically, there's an increasing interest in circulating tumor cells, like you can see here. This is the publications with CTC in the name. And now how to use them. We have a preparation set where you can have this blood tubes with 15 milliliters of blood. 4 to 27 hours at room temperature. You don't even have to cool it down or something like that, to our central lab in Bavaria in Germany with FedEx or DHL. It's a standardized version. It's called exempt human specimen, and you can use it very quickly without any problem. What we do is a semi-automated testing. It's an image evaluation that works on almost any solid tumor, but not on lymphoma or leukemia.
Okay.
This is how it works. What you see here is you see the circulating tumor cell within the bloodstream. And what we look for is the surface antigen. It's called EpCAM, epithelial human antigen. And we use simple staining with a fluorochrome labeled antibody, which sticks on that antigen on the surface of the circulating tumor cell. And with our microscopes, we can count this cell. That means we have a quantitative detection of vital circulating tumor cells. We don't need a fixation. We don't need any isolation, and we don't need any enrichment. Because fixation, isolation, or enrichment all mean a very huge loss of cells.
So my question is this surface antigen only on tumor cells? Could it appear on normal epithelial cells? Do you have epithelial cells floating in the blood also?
So you might have a little epithelial cells in the bloodstream. So if you cut your finger, for example, tissue cells come into the bloodstream, but at a completely healthy patient, there should be almost no epithelial cells in the bloodstream.
Okay.
So this is how the cells look like. And if you look at the cells, then you see there are cells with a very high staining, like this one over here, but even with a very low staining, like this one over here. And
in the left upper corner.
In the left upper corner. We do a double staining to see whether the cells are alive or dead. If the cells are dead or already dying, then the cell membrane is damaged, and another color, another staining can get into the cell core, and we see it on the microscope like a red dot in the core of the cell. So if the nucleus is stained red, then the cell is dying or already dead.
I see.
We can monitor the therapy success using circulating tumor cells. We do it almost every investigation of us is an adjuvant treatment. So we have 3 different types of treatment: success or not success. Successful is if there's a decrease in cell numbers by 10-fold or more. This is a normal chemotherapy, but it could be at any other cancer therapy. There are cells that do not react on any on the therapy at all, and the worst case is if there is an increase in cell numbers by more than 10-fold.
Forgive me a second.
Yeah.
You have to correlate what you see in the blood with what happened clinically with the patient. So if you give a radiation or another treatment that destroys a lot of the tumor and some of the cells kind of get lost from the tumor mass, go into the blood or whatever, the question of timing, did you find any interesting information from the timing of the response?
Yes.
Related to the clinic. And I would be very interested to get in the future from you some example of clinical correlation to imaging correlation. But I'm glad you give me this lecture so I could share it with viewers and they could understand the basics.
Okay.
But I already could see that privately we'll probably have too many questions and we will talk more in the future. So continue with your nice presentation. Go ahead.
So correlating with the clinical outcome is what we see over here. We see that this is a Kaplan-Meier plot used in scientific, very much in the European region. We see that these are the overall survival patients, and these are the days that we had this investigation on. And you see the green line are the patients with decreasing cell numbers under therapy. And you see that they do have a very good survival rate. The blue line had marginal change or no change at all in the cell numbers, and they went a little less good. And the red line, these were the patients where while therapy, the number of circulating tumor cells were increasing. And what you can see over here is that increasing cell numbers correlate highly significant with a very poor prognosis. So every step down is a metastasis developed.
I could see very well the graph and I see the colors. Could you go again specifically tell me what is the green, what is the red, and what is the blue? Or maybe in the other order. What is the green, what is the blue, and what is the red?
Okay, the green one is, if we go back, the green one is decreasing cell numbers by more than 10-fold under therapy. The blue one is no change of the cell numbers under therapy, and the red one is increasing cell numbers by more than 10-fold under therapy.
And let me interrupt you just a second to clarify one point. When you, on one test, catch number of cells, how many cells do you catch? Because in the regular CTC in the States, we talk about more than 5, less than 5. What are the numbers that you actually see?
So let me change to another presentation. This is a presentation where you can see a short comparison between Maintrack and the CellSafe or cell search system. Cell search tells you if you have 2 or 5 or something like that tumor cells in the blood, then it's a bad prognosis. But if cell search finds 0, we find already 2,500 events. And so we can say we are about 1,000 or 2,000 times more sensitive than most of the other methods used using enrichment or using any other technique.
That's very interesting, and it remains to be seen how it translates to the clinical outcome. But I would like you to continue with what do? You find the cells, you count them, you stop there. Is there any other investigation that you are doing on those tumor cells?
Sorry, yes. So if there's an increase in cell numbers, what can we do? This is the question you asked me.
Right.
We published this already. We do chemosensitivity testing. That means we drop in a day dose of possible substances, let's say substances. If we got increasing cell numbers, then we do chemosensitivity testing. That means we take a blood sample of the patient and we divide it into some parts, and then we drop in a day dose of the possible medications that the patient could or should get. So what we see is whether a cell dies under a drug effect. This is what we see. And we do counting as soon as we have put in the drug into the blood, and then we watch them in different time hours, whether one or the other drug kills more circulating tumor cells. As a patient, one of the first questions is, how much blood will you take from me? Because if you check for different things, how much blood is a usual sample? It's just 15 milliliters of EDTA blood, 15
okay.
milliliters of just normal blood.
I understand.
So what you can see here, for example, is we tested doxorubicin versus capecitabine. And what you can see here is that the doxorubicin worked much better on the cells. It killed almost, let's say, 80% of the cells, while in the same time, the other drug killed just 20 to 25% of the cells.
Right.
So we do also test different concentrations of the drugs to see whether there's a change of, but you can see 1/10 of this concentration of a day dose compared to the blood equivalent of the patient with, let's say, 5 to 7 liters of blood, or the 10-fold of the concentration, which would nobody get because it would be killing the patient.
Yeah, it makes it simple. But one question that I always like to be leave the patient with some impression. Is there anything you allow to tell me about the future? What do you intend to carry it even further, or at this moment it's still under the microscope, sort of speaking?
Okay, so there's one thing we do in science right now. We are taking this circulating tumor cells in the cells to growth. That means we are building up micrometastasis out of the body and test whether a drug works on this micrometastasis. And we found out that there are some drugs that work better on micrometastasis than they do on circulating tumor cells, or even that drugs that work on circulating tumor cells don't even work on that micrometastasis. We call them tumor spheres.
So what's very interesting to me, so the next thing in the future, you have more, you culture those cancer cells. And I have a question you may not know, I truly do not know because this is not my field. When you culture them in a plate or a test tube or, you know, in a very artificial in vitro situation. Mm-hmm. My question is there any culture done where you could take the soil? What do I mean is, we talk about cancer. The cancer is the seed, and the soil is the micro-tumor environment. Mm-hmm. Are there any attempts to take perhaps a preparation of, you know, ground, you take a bone, you grind it, you put some elements there, and then you put the cancer cell there, and you try to see what is the effect when it's more natural kind of environment for the tumor cells and not only in the test tube. Because in the past, I had experience with chemosensitivity. It was perfect in the test tube, not on the person, because there were different clinical situations. In the lymph nodes, so I wonder, I do not know, I need to learn more about cancer cell culture. If they have culture that means together with lymph node, fresh, or with bone, do you know if anybody is doing it?
I don't know if anybody is doing this because most of the other methods are not able to extract vital circulating tumor cells. We are, as far as I know, the only method that uses vital cells and not damaged or dead cells like they do on enrichment or on isolation or something like that. Okay. But we are trying to put these spheres, for example, into mice. This is one scientific project too.
I see. So that will be probably a topic for another conversation about instead of the circulating tumor cells, what do we know about spheres or about DNA or RNA floating in the blood? And I'm looking forward to hear from you again. And hopefully I can talk to your other expert in the company, Senior Pachmann, because I have an interest in sequencing circulating tumor cells and the information that we find from the sequencing to create adoptive cell therapy. But that will be a topic for another video, just to explain what adoptive cell therapy and what is sequencing. So I want to thank you very much for your presentation. Mm-hmm. You take care.
You too.
Wish you the best.
Over to you, all the presentation and publications.
Thank you very much for this very nice presentation, and I hope that the viewers could get some more information, and I hope that the articles mentioned on the slides, you make them available to me, preferably in a URL so I could put it in my video blog. I wish you the best of luck. You are a young man, so stay well, stay healthy, and I wish you the best.
Thank you very much.